So my title of my talk today is Identification and Optimization of Critical Raw Materials to Regulate Monoclonal Antibody Glycosylation. So what I'm going to do today is kind of explain to you our techniques and our systems and strategy and how we go about regulating glycosylation. And then I'll do a case study at the end with a client that we did some work on, and then hopefully explain all these things in good detail so you understand what we can do and offer for the corporation. So kind of an overview of what the industry is like. Basically, the biopharmaceutical industry is from development to manufacturing, and really from making that research cell line clone all the way up to the final drug product. This is kind of the area that SAFC kind of supports. These are all the products and capabilities that we do for the industry, media and key components, supplements and feeds, growth factors, sera, concentrated products. And then when you get down to the downstream side, we start looking at cleaning solutions, inorganic salts, purification buffers, reagent. This is all the components and systems that we have in place to support the industry. So we're a full capable company to support the whole industry. Today we're kind of represented by BioReliance, which is they have a booth in here. We don't actually have an SAFC booth, but we're here to support the BioReliance folks. And BioReliance, of course, is working with making sure your cell clones and all your systems are virus-free. So certainly visit them if you have a chance. So a little bit about media development services. Basically, we provide develop custom and catalog formulations that improve performance, reduce risk exposure, and provide assurance to product quality. So we interact with our customers in multiple different ways. I kind of want to explain that a little bit. The first level is really, we would actually send media and feed formulations to the customer. They would do the screening in-house, work with us, and we would actually ask what kind of cell line you're working with, and we would actually select media formulations that would be good for that cell line, because we have a lot of experience and a lot of history on that. The next level of interaction would be consulting projects. This is where we would really interact with the customer more directly. We would develop a strategy, we would develop experimental designs, and we'd understand the results out of that and queue for the next experiment and really help them do that development work. And that's really geared towards, if they can't send their cell lines to us, we'll help them in their facility. And then the last level is actually where the cell lines can come into our facility, and we can use our expertise and capability to really do that media and feed optimization in our hands. We're fully developed around doing that in our labs, so we can do it usually a lot more efficient than the customer can, because we have a high throughput process, which I'll explain a little bit. But typically, a customer would only have maybe, they can do maybe 20 conditions, where we can do many, many more. This is all really just geared towards meeting the target goals of their customer. We're very customizable. We want to work with our customers. We would pretty much understand what they need, and we would kind of mold ourselves into that to help them out. So I'm going to kind of talk about and describe our next generation of media development. And this is really the ability to identify critical raw materials to meet the goals. So what we want to do is understand what each component in that formulation is actually doing and what kind of response you're getting out of it. And I'll describe the tools and how we go about doing that. If you look at our capabilities, we have a very large chemically defined library. We have our high throughput technologies, and we also do a lot of statistical analysis, mainly around DOE and MVA, multivariate analysis. So it's there's a big bevy of tools that we can utilize to really get the media formulations and feeds for our customers. Just describing our media and feed library, I think Stuart mentioned he had a toolbox. I think we have maybe a Snap-on drag-in toolbox. I don't know. So we have a very large media library in our house that, over the years we've developed, figured out, and utilized that media and feed formulations for our customers. So what we've been trying to do is really analyze that media panel that we have, and by using statistical analysis, mainly MVA, you can actually start looking at the media diversity set within those formulations. We have a lot of formulations, maybe one or two components have been moved, but what we want to do is understand that. And by using statistical analysis, we can start looking at the hierarchical plot of understanding how similar those formulations are to each other. and utilizing that knowledge to really do a very specific media and screen projection of how you can interact with those media and feed formulations. So we take that historical knowledge and the understanding of our media formulations, and we can select specific media and for a specific cell line. We've been working with a lot of customers, a lot of cell lines in the industry. We got a lot of history with that, so we can start projecting what we should be doing already up front. And this is really geared towards to really reduce time and cost associated with that development project. That's what my goal is within this group, is to really reduce the amount of time it takes to get you to an optimal formulation. So, and one of those techniques is actually using high throughput screening methodologies, which I'll talk about. So this is a kind of a pictorial of our high throughput systems. We have a biomech which is really set up to do 96 deep well plates. This is our liquid handling portion of it, so we put the media, the feeds, or whatever is going on into those wells through this high throughput system. We have a TomTech, which basically can inoculate the cells into those 96 wells, and this also does dual, where it pulls out samples for analysis. Once we get the plates inoculated, they're put into the microtron, which is a high throughput 96 well plate incubator. And the way we designed this is actually, it took us a little bit to prevent the liquid to being evaporated in those 96 wells, because you have a small volume in there. Over time, that's going to evaporate, so we had to figure out a sandwich module to put on top of it to keep it from evaporating and to get good oxygen exchange into those cells. So this it's a high throughput system. Of course, we can do quite a few conditions in that. Once the cells are incubated, pull samples out. Those samples will be looked at for our benchtop flow cytometer. We use the Hypercyte with the Accuray flow cytometer. And samples will also be sent over to our Forte Bio for IgG analysis. These are typically MABs we're working with. And we also have a very good analytical capabilities. This is a picture of our analytical R&D lab in our St. Louis facility, and where we'll talk about our mass spectrum analysis, our capabilities there. So we can look at protein quality. So chemically defined media and feed selection. So what we want to do is basically identify media formulations that are good to go forward with. So we want to identify a media and feed target for optimization, establish culture response in diverse ranges, identify raw materials and correlate the cell culture performance with those raw materials. So historically what we would do is basically pick these top two formulations and move on, right? They're doing the best, that's ones you go forward with, right? Well, we're missing a lot of information down in here because each one of these are a different formulation. From this plot, you can see the control is right here, and when we do this diverse formulation screen, you get a lot of data here. So to really utilize that data, we developed using multivariate analysis. So now we can take a look at all that data set in there and understand from every component and every condition what that effect is, right? So we can look at positive and negative correlations and statistically analyze that data set. And we can look at any response that is out there, right? So specific productivity, doubling time, waste product formation, glycosylation, which I'll talk about a lot today. Any client-defined response, this system can put that in there and we can analyze it. So what are the therapeutic critical quality attributes? I'm kind of embarrassed to put this slide up here after Mike's talk, so I'll go through this a little bit lighter than he did. We have charge variant, glycosylation, immunogenicity, aggregation and degradation, and primary and higher order structure. A lot of these will be done with the cell line development, so what I want to do is focus on basically the glycosylation. So again, this is my cartoon of what Mike probably did a lot better than I would, but this is the ER as it's made in here. It goes to the Golgi apparatus, and basically you get maybe 4 major glycol profiles that we'll look at. So mannose, G0F, G1F, and G2F, as you'd seen before. So what we have developed is a high throughput method for looking at these samples. So we have an intact mass electrospray ionization assay where we take a protein A purification in a 96 well plate, and we basically look at the raw data, reduce the MS, deconvolute the data, and you basically get intact mass data analysis with the G0, G1F, and G2F. We like this methodology because, one, it's fast. We can throw a lot of samples through there. We can get analysis within 3 to 4 days of multiple plates. So we can start using this information when we're doing media optimization or media feed optimization. So it's a very high throughput methodology to get you answers that you can utilize when you're doing the optimization screen. So if we look at chemically defined media and feeds again, we're identifying lead media and target optimization. So if you look at these two media, there's basal media 1, basal media 2. This is the same cell line. These are the same feeds. They're not quite in order because there's two different responses we're looking at here, but you'd want to probably start with basal media 2 if you wanted to have an increased higher order glycoform, because all these black lines are G0, and you can see that the G0 is much less than this one, and we're looking at G2 increase in basal media 2. So it's very important to start out with the right basal formulation, because to overcome these in this basal media 1 would be a lot more difficult than if you started right here if your endpoint is a higher order glycoform. I want to talk about our case study. We want to incorporate this information into protein quality to design. This really increases our design space to customize media and feed formulations. So we want to optimize components ranging that balance titer with product glycosylation. So now we're going to talk about how we actually did that. So the objectives and strategy of this, we did a proprietary screen, identify top chemically defined formulations for targeted optimization, generate culture responses with diverse ranges, identify critical biochemical components within those ranges. The targeted optimization is maintain titer with increasing product glycosylation, and the higher order was G2F is what was most interested here, and characterize the critical biochemical components, and also wanted to reduce risk exposure by removing the plant hydrolysates within this feed and this particular customer's product. So here you see the viable cell density plot. The black line is basically the control. This one has the hydrolysates in it. We had a control of a CD formulation from our bevy, and then we did optimization of that particular formulation. You can see the response we get. You see a high and a low response, so that means that we can start determining maybe what's causing the high and low response based on this information. Again, here's the volumetric productivity. Again, you can see where the black line here is control. Yeah, we're getting 1.6 grams per liter, but most of that is not usable protein. It's not good protein quality for what they're interested in. So again, we see that there's a wide range of productivity, so we can start utilizing that information to help us in predicting the next round. So this is a typical printout for multivariate analysis when we map the culture performance. All these purple dots are basically all the raw materials. Now you got to remember, all those raw materials were out of those 56 conditions, right? So you've seen all the impact over those 56 conditions. How do the response and the general response look, right? So this is a principal component response map. And then all the yellow ones are actually the responses or the outputs that you're looking at. So if you're looking at G0F or viable cell density, how you understand this is anything close to that yellow dot, any purple dot close to the yellow dot has an influence on that particular response. Vice versa, if this one's having an input or influx on the viable cell density, it's also making G0F decrease. So what you want to do is take raw materials that are close to that, and those will be important ones to look at. So if you take a look at this next graph here, this is another way of looking at information. This is what's called a variable importance of projection. What we want to do is look at all those raw materials again. Now we can start lining them up for importance from most important to least important. And typically we want to look at things over a value of 1. It gives us a little bit more targeted area to actually focus in on. And again, these could be positive or negative. We don't know the importance of it if it's positive or negative at this point, but it has a strong importance on the actual result. Vice versa, we can look at what's not responding, and if these aren't responding or having any kind of effect on the outcome, you can actually start taking those components out of the formulation as long as they're not, needed. You need to have salt in there and stuff, but it's still. So you would actually start doing deficiency testing on these and taking them out, and that actually would reduce your risk exposure to all these raw materials that we're sourcing those, we're testing those. If you don't have to source and test those, then they're not. You can take them out and you can just reduce your load on the actual final formulation. So this is the strategy we use for this particular project, the targeted optimization. Once we had the targeted formulation, we want to make sure the formulation is streamlined by using deficiency testing and component consolidation. While we're talking about component consolidation, if you have like iron in there's a couple different forms of iron, you maybe want to put those into one form of iron. And then we would proceed to design of experiments. We could do a fractional factorial or essential composite designs. These are all incorporated. Now that you know what components to look at, you can start putting them into these focused DOE designs and start really nailing down what components have and what kind of response. Then we have usually these formulations made by our immediate advantage group. This is a small-scale, non-GMP resource that we have access to. We can also look at stability testing those formulations. If we range components real high or real low, do they have any response for, if we're talking about a 2-hour life study with the Stuart was talking about, we can test that out early on and make sure that we're not having that same kind of problems. And then we confirm that performance of all these formulations. So within this target optimization, the goal was to really increase G2F, right? So the ultimate results here, this is the control. The peak viable cell density was 10.7. That was the initial control. The optimized CD formulation was 13.7, so we were slightly able to increase overall growth. Peak titer was 1.7. The control is 1.6. The optimized was 1.81, so we were able to slightly increase peak titer. But what was most important was we took it from 1x G2F to almost 3.5x G2F, and we're able to meet the innovator. This is a biosimilar, so we were able to meet the innovator glycol profile with this particular subject. So we were successful in chemically defined formulation, no hydrolysates, takes out that variable, increased growth in titer. We increased 3.5x of the higher order glycol forms in compared to control. So very successful project. This is kind of just a description of, based on all this information that we've learned by being able to use this high throughput methodology, working with a lot of customers, working with a lot of different cell lines, we were able to develop a new product called Excel Glycosylation Adjust, or GAL Plus for short. And this is a protein quality supplement. You add it into your media formulation and you get a positive response, or it will actually increase G2F. And it does so well that you probably need to titer it in a little bit. If you put too much in, you'll over-glycosylate your protein. It's a novel, ready-to-use GMP concentrate, and it has a positive response for glycosylation or higher order. If you look at this graph here, you can see we have the chosen in the DUXB11 and CHO-M. If you look at G02F, basically you don't see any kind of blue lines here. This is the G2F. And all these, after you add it, you get a marked improvement on G2F higher order glycol forms. So the product's available. You should check into it. Just as far as acknowledgements, this is all the group that I work with. My director is Bruce Lear. Senior director is Kevin Kaiser. We have a media development service in St. Louis. We have an analytical in St. Louis. Raw material characterization group is in Lenexa, Kansas. We have a chemistry excellence team in Lenexa, our immediate advantage, and this is a group of all the folks that we're working with. So with that, I will take questions. Thank you.
I'm Sean Eckerly with Patheon Biologics in St. Louis. You guys are doing a ton of work here on your side to help support a customer.
Yes.
But what happens when you've done all of this successful work in your hands, give it to the customer, and now it's not performing the same way that you're seeing here?
Typically that doesn't happen, but it certainly does happen occasionally. What we find is the culture system that the client is growing in is maybe slightly different than what we developed that formulation for. So we would go back and take another look at it. And if it's a unique system, we would try to incorporate that into our model, make sure that we're not missing something. So typically we'd try to go back and take another look at that.
Thank you.
Welcome. Hi.
Quick question. Sorry, I missed the beginning of the talk. I just wondering, is this such big amount of work? Do you use the high throughput system?
Yes, I can go back up to it. So it's a 96-well based platform that we use. This is the system that we have. This is a liquid handler, TomTech incubator, and then looking at the actual results of either growth and viability or protein production.
Okay, so regarding analytical support, also the high throughput.
Yeah, so the high throughput intact mass analysis we use, and they also do spent meat analysis for us also.
Okay.
So we have a lot of capabilities. We are a chemical company, so we have the ability to look at a lot of information on any component that's in these formulations.
Great, thanks. And quick question about the timeline. Let's say for the work we talked about. Roughly how long does it take?
The timelines are usually regulated by the customer, right? So if they have, and we do need to educate customers sometimes. You can't do things within a month. I mean, cells do take time to grow. So these projects, typically we'll lay out a whole media development project, probably be 8 to 9 months. It depends on what the outcome is and what they're looking for. If it's just a feed, we can probably do that in 3 to 4 months. So it just depends on what they're looking at. Okay. But going from basal formulation to feed formulation all the way through the whole process, probably 8 to 9 months. I'm always pushing our group to make it shorter.
Okay, so. Thank you very much.
Thank you. All right. Thank you.